Hello, everyone. Thank you for attending and welcome to today's live webinar, Novel Tools for Translational Research, Ex Vivo Human Models for Advancing Pain and Itch Drug Discovery. My name is Diane Royer. I am the Marketing Manager at Neuroservices Alliance, and I will be moderating today's webinar. This event is part of a series that features our partners, neuroscientist experts, and showcase their work to show you how Neuroservices Alliance can be your efficient CNS drug discovery partner. We know that your time is valuable, which is why our online events all follows today's format, 20-minute presentations, followed by 10-minute questions and answers. If you have any question at any time, don't hesitate to submit them in the question box that is located on the bottom of the GoToWebinar panel. I would like to inform you that this session will be recorded and you will receive the recording after the live session. However, I strongly encourage you to stay until the end, as it is the occasion to have our speaker, Ghetti, answer your questions live. Before starting, I would like to say a few words about Neuroservices Alliance. We are a consortium of CNS CROs specialized in vitro and in vivo electrophysiological and behavioral studies. Our goal is to deliver endpoints facilitating decision-making to our clients for their CNS drug discovery programs. We value direct and efficient interactions with our clients, and we rely on a team of more than 50 PhD scientists. As a speaker, we have Dr. Ghetti with us today. He is the Chief Executive Officer of Neuroservices Alliance member, AnaBios Corporation. Hello, Andre, and I will let you take the lead from here.
Great. Thank you, Diane, for the introduction, and thank you for the opportunity to present the work that we do at AnaBios, and thanks everybody for tuning in today for this presentation. So what I will do today is briefly provide some context, discuss the translational challenges in drug discovery and development, the novel translational paradigm that we have developed at AnaBios, and then show an example of how this can be applied, and I will discuss specifically an example for pain drug discovery. So I'm sure you're all familiar with the high attrition rate of drug development, where less than 10% of molecules that enter clinical trial actually reach the market and patients. And this high failure rate is typically attributed to the challenges in translating preclinical animal data into clinical human outcomes. And so the question is, what are the data that we're missing? What is it that we should be able to learn at the preclinical stage in order to bring in the clinic drugs that have a better probability of success? And there are really three main areas that we think are critical and on which I will focus today. First of all, we really need to be able to accurately predict the risk-benefit ratio of drugs already at the preclinical stage. So having a good safety and efficacy profile that is predictive of human responses. But this is really not sufficient. We need to also be able to understand what specific patient populations could benefit from a particular drug. And so this is critical because oftentimes, as I will show in one of the examples, we may have a great drug, but if we select the wrong patient population, we may fail to meet the end goal, the endpoints of the clinical studies. And the last point is the dosing. Oftentimes, the potency of a molecule in animal models is quite different from the potency in human. And therefore, if we have to rely on animal data for setting the dosing in the first in-human studies, we have a high risk of either over or underdosing patients. And here the risk is that if we overdose, we're going to see a lot of side effects and adverse events. If we underdose, we may not see efficacy. So the strategy that we have developed at AnaBios is illustrated simplistically in this slide. And the concept is to bring to the laboratory for preclinical studies human organs, human tissues, and human cells that can be used for assessing drug action in the context of human biology and pathophysiology. And we do this with novel methodologies that allow us to maintain full viability and functionality of these samples in the laboratory so that one can indeed perform studies that provide predictive data of clinical responses. This is accomplished by working with a network of hospitals and transplant centers across the country. This is something we have established over the last decade. And we also have developed very unique and proprietary methodologies for recovering organs and tissues and preserving the viability during shipping from all these various centers to our laboratory in San Diego. So we receive organ and tissue which are in full, viable, functional state, and we can use them right away either for establishing cell isolation and cell preparations for cell-based assays or conducting measurement and assays on tissue preparations. We can also bank these tissue samples so that they can later be used for gene expression analysis, proteomics analysis, or other type of biochemical investigations. So the use of human tissues in research is not novel, but what we have done have been able to really scale it and also standardize using optimized, dedicated methodologies that specifically ensure the viability and functionality of the samples. And this is really novel because both scale as well as quality and consistency of tissues is something that was missing and is critical for drug development applications. So over the years, and we've been doing now this for almost 10 years, we have worked with numerous pharmaceutical partners over the US, Europe, Asia, and have really seen how this approach can be predictive of clinical outcome. It truly lowers development risk due to interspecies differences. And one aspect which is extremely important is that it also enables the study of drug actions in the context of pathological states, which is really the most interesting point in the context of drug discovery. So we have the ability to recover tissues not only from donors that were not affected by any particular pathology, but also donors that were affected by specific pathologies of interest. So if, for example, we're conducting studies in the context of heart failure drug discovery, we have the ability to bring to the laboratory hearts from donors that were affected by heart failure. And last but not least, we have seen a very strong correlation between the potency that we measure in these ex vivo human preparations and the clinical responses and the clinical dosing. So this quantitative aspect of this particular paradigm is also extremely important and critical. So I want to give now an example from the work we have done in the context of pain drug discovery. And as I'm showing on this slide on the right side, pain is really a universe of conditions. So there are many different mechanisms and pathophysiologies that result in pain states. And these different pain states manifest with different sensory modalities and really have underlying distinct mechanisms. So most likely they will need to be treated with different type of drugs. Now, traditionally, pain drug discovery has relied on animal models, in particular rodent models for pain. And there are at least 15, 16 different pain models going from the injections of irritants into various parts of the animal's body to nerve ligations or other type of nerve injuries. And the real problem is that we don't have any idea of how these different rat or mice pain model map onto the human pain conditions. And so it is impossible to know if your drug is working in a particular set or subsets of these pain models, which particular patient populations should be targeted for the clinical studies. So what we do in the context of pain drug discovery is to recover the dorsal root ganglia. These are the ganglia on the side of the spinal cord that house the cell bodies of the sensory neurons. These are the neurons that detect injury and dangerous conditions at the periphery of the body and send pain signals in the form of electrical impulses, action potentials to the spinal cord, and then from the spinal cord, the signals go to the brain where we perceive pain. So we can, from the ganglia, isolate the sensory neurons, put them in culture, and use them for physiological functional studies, primarily using electrophysiology or other imaging methodologies. And so we can, for example, measure in these cultures the electrical activity, the very same impulses that are responsible in vivo for transmitting pain signals from the periphery to the central nervous system. And so, for example, the ability to study the modulation or the inhibition of these pain signals is really something extremely valuable for pain drug discovery. Now, we can do this by either measuring the activity of drugs on cultures that are maintained in a basal, quote-unquote, normal state, or we can induce pathological states in the culture by adding, for example, inflammatory agents like prostaglandin, or exposing the cultures to agents that are known to induce neuropathies like chemotherapy agents like oxaliplatin. And so we can see in the cultures physiological and phenotypic changes which actually correlate very well with the type of pain that patients experience when they either have inflammatory pain states or chemotherapy-induced neuropathy, for example. So we call these artificial in vitro pain models, and these are a first strategy, a first step for very conveniently starting to identify drugs that have activity in the different pathological states, and also distinguishing drugs that may be more active in the inflammatory state or in the neuropathic state. And then the next level of investigation, which is really the gold standard, is to recover dorsal root ganglia from donors that were affected by one form or another of chronic pain. And as I'm showing in the lower left part of this slide, approximately 20% of the cases that come our way are from donors that were affected by one form or another of chronic pain. And on the right side, at the bottom, what I'm showing is a recording of spontaneous activity in cells obtained from a donor that was affected by a form of chronic pain. I think it was knee arthritis. And this is very unusual because typically these cells are extremely quiescent in culture, and we see these levels of spontaneous activity only in particular states of pain. So these kind of preparations obviously can be highly valuable for prioritizing compound and identifying the potency of the compound and the ability of the compound to inhibit this particular pain state. Now, in one example I want to discuss is the case of a sodium channel blocker. This is a new generation sodium channel blocker. Was originally discovered by a company called Convergence. Later, the program and the company were acquired by Biogen. This is a sodium channel blocker that inhibits quite effectively action potentials in human sensory neurons. And as I'm showing at the top left side of the slide, the inhibition of action potential was actually very good under normal nociceptive conditions. However, when the neurons were exposed to inflammatory agents, as you can see on the right side, the ability of the compound to inhibit action potential in sensory neurons was drastically reduced. Why is that the case? Well, it is known that under inflammatory conditions, there is a switch in the expression of different sodium channel subtypes. And given the selectivity profile of this particular drug, the channels expressed under the inflammatory states were not blocked by the drug anymore. Now, we also tested this compound in DRGs isolated from a donor that was affected by low back pain. As you can see, there's really relatively little inhibition. And by the way, the green area in these graphs shows the 30% or more inhibition of action potential. This is something that we have empirically established using a number of clinically used analgesics. This is the area in which inhibition would be predictive of clinical analgesia. So based on these data, we predicted that waxatrigine would not be particularly effective or would be not effective at all in inflammatory pain states. And one case that has plenty of inflammatory components is low back pain. And in fact, the clinical translation really matched extremely well these predictions. So waxatrigine had positive data from an early trial in trigeminal neuralgia. Trigeminal neuralgia is a condition in which the trigeminal nerve is compressed by a vessel. Is not really a neuropathy in the classic sense. There is no real damage to the nerve, but the compression causes the activation of the nerve and the pain. And so we think that the normal nociception activity shown by waxatrigine in this example explains well its activation and activity in trigeminal neuralgia. On the other hand, a second trial specifically for low back pain failed to meet primary and secondary endpoints. And again, this is not surprising given the inability of this drug to effectively block sensory neurons' action potentials in inflammatory pain states. So this is an example of how a compound could be effective in particular patient populations, not effective in others, and how these ex vivo human cells or tissue preparation could really help tease out these subtleties. A brief mention about itch. So sensory neurons are not only responsible for pain sensation, but also itch sensation, and there are many forms of chronic itch which are really problematic and have been difficult to treat. So the sensory neurons in the cultures are responsive not only to stimulations by algogenic substances, pain-related substances, but also pruritogenic substances. And so we're showing some examples here with histamine and chloroquine that are able to elicit action potentials in the cultures. And on the right side, actually the middle part of the slide, what we're showing is that activation of different non-histaminergic itch pathways in the neurons is possible, and these responses can be sensitized, for example, under inflammatory state. The other thing that this slide shows in the two pie charts at the bottom is that by applying a number of stimulations to the same set of cells and recording the responses to the different pruritogenic substances, we can create a fairly comprehensive and complex phenotypic map of the cells in the cultures. And we can also show how this phenotypic map changes in a pathological state like inflammation. Now, this is important because, again, if you want to target a particular pathological state, it is important to understand what changes in that state and what are the most appropriate targets for treating that particular itch conditions. And I think this is really the essence of what I wanted to show this morning. So I really wanted to deliver the message that the new AnaBios paradigm enabled the assessment of drug effects ex vivo in human tissue and cell models. This is enabling the ability to study compound action, drug action in the context of pathological states, therefore predicting the potential in the clinic and enables the selection of the most appropriate patient population. And the potency, the quantitative aspects of the data that are generated by this platform is also extremely important because it's extremely valuable guidance for dose selection in the first-in-human studies. So overall, this is an approach that truly maximizes the opportunities for successful drug development. So I will stop here and open up for questions.
Okay, thank you very much, Andre. We do have some questions. The first one is, How did you differentiate different types of DRG neurons and monitor their response?
Yeah, that's a great question. So, as I mentioned, we are using both electrophysiology as well as imaging techniques. So these are all live experiments, and during a test like this, we typically use various type of controls, agonist or antagonist. So if we're using current clamp recording of action potentials, we can look at the responsiveness of the cells to a particular agent that inhibits one subset or another of sodium channels. We can monitor the ability of cells to respond to capsaicin or other agents. So we essentially do a functional phenotypic type of profiling on the side of the test, which enables us to characterize the cells we're recording data from. We also have the ability, and this applies especially to the imaging type of studies, at the end of the experiment, we can fix the cells and then perform immunostaining for a variety of cell markers. But this is a great point because it really gives me the opportunity to mention that there are at least 20 to 25 different sensory neuron types in these preparations. And what you can see is that oftentimes drugs are effective only on a subset of cells. And so understanding the subset of sensory neurons on which the drug is most effective can then provide useful data for deciding which type of pain the drugs may be most appropriate for. For example, if you see a drug that is mostly active on heat-sensitive sensory neurons, well, all of a sudden, all the pain conditions in which the main complaint is a burning sensation becomes a very good target population. And this is unfortunately something that is missed, for example, in preparations that are derived from stem cells. So stem cell-derived sensory neurons typically have phenotypes that are not really matching the true phenotypes of the human sensory neuron population. Not all phenotypes are represented, and there are many descriptions of stem cell-derived sensory neurons that seems to be a mix of different neuronal populations. So this is really one of the benefits of using primary cells.
Thank you. Now, how long are the human sample viable? Are you able to collect meaningful data from the human tissue samples over an extended period of time, days, weeks, or more acute, hours? Specifically interested in DRG, brain, and spine.
Yeah. So the DRGs are, and again, these are primary cells. So for the most part, they don't divide. We don't propagate. These are essentially plated, and they're viable for, the DRGs are viable actually for months. But we have seen that they are phenotypically stable for the first 2 weeks in culture. So we typically don't use them beyond 2 weeks in culture. Spinal cord is actually a new frontier for us. We started recently in a collaboration with Eli Lilly, which we publicly announced earlier this year. And we're currently working with spinal cord slice preparations, conducting both imaging as well as we're developing now electrophysiology-based assay. And we're establishing organotypic spinal cord cultures. And so we're now able to maintain viable spinal cord preparations for up to 7 days. Brain, we don't have yet that ability, but it's coming up. What we have learned from working with spinal cord preparation is going to provide us the knowledge and the guidance we need to develop a brain slice preparations, which I think will be coming later in 2021.
Thank you. Now, have you tried opioids in human DRG neurons in inflammation or low back pain?
Could you repeat? I didn't understand the first part of the question. Have we tried
opioids?
Oh, yes.
In
so
human
we have tried. Yeah, we have tried a number of opioids. We've looked at opioid direct responses. We've looked at opioid induced calcium signals in the neurons, and we've also looked at the modulation of sodium channels by various opioid pathways. So we actually published, there was a poster that we published with Eli Lilly a few years ago, so that should be downloadable from our website.
Thanks. Now, can you compare human DRG to Rodan DRG in the same setting to provide translational info?
Yeah, we have done some of the works, especially early on with some of our early clients, and now we don't do that routinely. Typically, partners come to us already with DRG rat or mouse DRG data, and essentially what we see is that the correlation or the translation is somewhat variable. So for some pathways, there seems to be good conservation. For other pathways, the conservation is not so good. In particular for itch, there are very significant divergences between the two species. But even for pain pathways, it really varies and depends. And also what seems to be different is the response to pathological states or pathological sort of stimulation. So it's really a mixed bag. And the problem is that it's difficult to know in advance what is going to be tightly correlated or what is going to be divergent.
Thanks. Are you able to isolate and use associated cells such as microglia and invasive monocytes?
Yeah, so the cultures that we establish are not enriched for neurons. So we try to culture and maintain in our preparations all the cells that are in the original ganglia. There's definitely plenty of glial cells, microglial cells. We have not done extensive characterization of microglia in particular in these cultures, but we do have evidence that those cells are there. We've also done experiments in which we add back to the DRG cultures macrophages or activated macrophages. The reason why we do this is that there is more and more evidence that in many chronic pain states, macrophages actually invade the dorsal root ganglia and really help establish a high level of inflammation. So these are all possible approaches to these questions.
Thank you. Next and last question for now. How are you able to lower temperature of neurons to show increased activity in oxaliplatin exposed preparations?
Yeah, so we have perfusion systems that are temperature control. So we have the ability to increase the temperature of the buffer we're perfusing on the cells, or lower, we can go down as low as 4 degrees. And that's how we've built these responses to cold in the neurons. And we clearly see that after the challenge with oxaliplatin, the threshold at which cold-sensitive neurons respond is increased. So what was originally perceived as cold at 15 degrees centigrade, after the oxaliplatin treatment, the threshold is closer to 18 to 20 degrees centigrade. And this is very interesting because one of the main complaints of chemotherapy patients that experience the neuropathy is cold allodynia. And so this matches very well the clinical setting.
Thank you. We now have some additional questions. Similar to the previous question, can you say some words in terms of interspecies differences? Any example of drug candidates for working in rodents but not working in human, or in reverse way?
Yeah, so situations of drugs that were active in rats or mice in particular and didn't show any activity in human, those are very common. Many of these, in the cases in which they have been actually advanced into the clinic, the clinical data really confirmed, and we're completely in agreement with our findings in ex vivo preparations. And it could be that in some cases, there's complete divergence in the pathways. One target that is central to pain transmission in rodent may not have that equivalent role in human. In other cases, we believe is really a potency effect. And so not having good information about the dosing in human, because all the potency data are coming from rodents, could really mislead and cause, in many cases, underdosing in clinical studies. The drugs did not work in rat and worked in human. Yeah, there were a couple of those. Although in one case was potentially was a mixture of possibly divergence in terms of target function, but also PK-related issues in the rodent. But yeah, we definitely have seen those examples as well.
Okay. Last question for now. Have you compared responses from non-human primates and human samples? If so, is the variability you mentioned between rodent and human still observed?
So we have not done side-by-side comparison between human DRGs and non-human primate DRGs. However, we have been working with pharmaceutical clients that have used internally the non-human primates, and it is a similar situation to the rodents, somewhat surprisingly, because from an evolutionary perspective, the expectation would be that non-human primates are a lot closer to human, and yet there are still significant divergences. So it doesn't seem to be the case that moving from rodent to non-human primates is actually improving translation a lot. I think it may ameliorate the situation for some pathways and worsen it for other pathways. So no really major advance.
Now, is there sufficient N from same patient types to show similar responses, or is there a lot of variability between patients?
Yeah, that's another great question. So if we are using the normal or say non-pain cells from donors that did not suffer from pain, there is relatively little variability. It may depend a little bit on the pathway. So sometimes on the itch side, there's a little bit more variability, but not on the pain side. When we start working with pain tissue, there's definitely differences between the pain tissue donors and the non-pain tissue donors. And among the pain tissue donors, it really depends on making sure that we are comparing, apples to apples. So we cannot look into, a low back pain sample and comparing with DRGs from somebody that was suffering from post-herpetic neuralgia or rheumatoid arthritis or another condition. So once you match as closely as possible, then the variability is drastically reduced. The other thing that we do, since these samples are very well annotated, we can know, for example, which specific part of the body was most affected by pain and do the isolation of neurons from the particular dermatome levels that was presumably affected. And so this really maximizes the homogeneity in the response. But overall, when you're doing fair comparisons, either between the normals or between the pain states of the same type, the data are very consistent. So we usually run in the early stages for programs that are relatively early stage. We can get extremely valuable information already from measurement in a single donor. And as the drug advances or as we approach clinical candidate selection, then it can be appropriate to look at least 2 or 3 different donors. But usually it's not necessary to run the measurements on more than 3 donors.
Thank you. Also, do you have data on gene expression or IHC from the DRGs?
Yeah, so we have data. I think actually the best data that has been published on human DRGs comes from the laboratory of Ted Price at Dallas. They have used, for part of that study, actually, they've also used DRGs obtained from AnaBios. And I would actually really recommend that you take a look at those publications. Those data are publicly available and really provide a fairly comprehensive overview of gene expression in these cell types.
Okay. Any validation set with drugs efficacious in the clinic for neuropathy, and duloxetine, for example?
So no, we have not. I don't think we don't have any data on duloxetine. We do have data on carbamazepine and some of the other anti-epileptics, which are also being used or have been used for pain. We have looked at some of the opioids. For some of the drugs that are currently used, one also has to be mindful of the specific mechanism. So if there are drugs, for example, that are acting at the synapse between DRG neurons and the spinal cord, well, actually that part of the pathway is not currently represented in our system, because I mentioned we have spinal cord preparations, but we haven't yet created the dual DRG spinal cord co-culture that actually would contain the synapse. So with the caveat of specific mechanisms, which in our case at this point need to be purely peripheral, yeah, we have a good number of additional data that I haven't shown and that we can share. I think a good amount of information is also available from our website.
Okay. Last question. AnaBios is a US corporation and therefore able to pair with companies using SBIR program US funds. Is that right?
Absolutely. Yeah, we are the recipient of SBIR NIH funds. We have successfully applied for grants aimed at development and validation of these methodologies, not only in the context of pain, but also for cardiac drug safety assessment, cardiac drug discovery, and so forth. We were also awarded a special grant late last year from the HEAL initiative. The HEAL initiative is an NIH initiative focused on addressing the opioid crisis, and they did recognize our platform and assays as an innovative approach to drug discovery in pain. And so in that context, I would say that we also have been collaborating with several academic laboratories, and we have been co-recipient and co-applicants on grants together with academic laboratories. This is actually a very good way for us to fund collaborations with academia.
Great. Thank you. Do you have any data from heterozygous humans for nociceptive associated NAVs?
So if the question is data from individuals that were carrying specific mutations known to be associated with pain, that's the way I understand the question. Mm-hmm. The answer is no. No, we don't. A lot of that work has been, I'm sure the person asking the question is probably aware, but the groups of Steve Waxman at Yale has done a lot of that work and published great stuff, also on human DRGs from individuals affected by those pain mutations.
Okay, thank you very much, Andre. For now, we don't have any more questions, so I suggest we finish the webinar. Thank you very much, Andre, for this very comprehensive presentation, and thank you everyone for your attention. You will soon receive a follow-up email with the full recording of this session and also contact information if you want to follow up. Don't hesitate to visit the Neuroservices Alliance website if you want to register for our next webinars. And after closing this window, a short survey will pop up in your browser. Please feel free to share your feedback. We appreciate it. And you can also ask any additional question you may have. Thank you very much. And we look forward to seeing you at our next online event.
Thank you all.